that you have acceptable level of the video, because we got some feedback from one participant that he was not able to see the presentation. So could you please just shortly confirm that the video is okay? Okay, thank you for your feedback. Probably that was a problem on the side of the participant. Thank you very much.
Hello, Amit.
Yes, Alvin. Can we start? Hello.
Hi, Amit, once again. I think we may proceed.
Okay.
Hello, Amit. Hello, Amit.
Yes, yes. Yeah.
If you don't mind, Amit, can I just show something quickly?
Yeah, sure.
In case some of you doesn't know about ESCO Pharma, I can tell you that we have grown a lot, especially now. The requirements are very, very active with COVID-19. Some of our customers, and we do many kinds of isolators for GMP customers. We cannot share this directly, but we can show you. Okay. Some of these companies you already know, Lilly, Mylan, we have Boehringer Ingelheim, we have Lonza. Okay, we have many applications here. Myra GTX, Teva, Salonic, yes, Russia. We even have our customers. I think our proudest one actually. We have customers in USA. FDA is our customer. So, we're talking about FDA recommending GMP guidelines. FDA is our customer. We have customers in cell therapy. We have customers in powder processing. Big names like GSK, like Pfizer. Pfizer. They are having a good time now. Many kinds of customers. We also sell to everywhere. South America, I think so. Yeah, we are present worldwide. Okay, everyone. I can't continue for so long. We are on tight schedule. Amit, that's all. So please take over the screen, Amit. Thank you.
Okay, good morning, everyone. Dimitri, my slides are okay? It's projecting?
Looks great for me.
Okay. So in this session, we'll be looking at ESCO Aster VaxiXL presentation. So here we will focus on the bioreactors and the bioprocess tools we have. So something on the introduction of the company. Everyone in this group is aware about ESCO when it was founded. That was in 1978. And that was the year when we started the group company. And ESCO began to pioneer the clean room technology during that time in basically in Southeast Asian market. And ESCO was mainly established to provide clean room air solutions that time for high-tech industrial and life sciences industries. And in 2000, when the focus shifted to life sciences more, then during that time, ESCO was very successful in adapting along with the wave of life sciences and biomedical sciences and healthcare. So the company has earned a reputation for providing and mission critical life sciences tools, as we are aware, in the lab equipment and also biosafety solutions, which can be seen in the ESCO Pharma. As you can see, we are into containment equipment. And in the year 2001, we got our first certification. This was actually, we were the first Asian manufacturer to earn EN 12469 certification, and that was for the biosafety cabinets. So we have come a long way from 2000 and 2001, and we also achieved our first NSF American ANSI certification in 2004. So if we talk about our employee strength, we are just over 1,200 employees globally, and we are a mix of scientists, PhDs. So you will see that what is ESCO Aster. I'll be detailing on ESCO Aster, which is one of the companies under ESCO Group. So this is how we started and where we are. So ESCO Scientific is mainly into lab equipment, as we all are aware here. Main equipment is biosafety cabinets, CO2 incubators, and all the life science equipment which you can find inside a lab. Now we are a major player in the market, and in many strategic markets, we have defeated our competition in a big way, and we are growing fast. And we are now one of the favorite among international agencies like United Nations and WHO. So we are a preferred vendor for them now. In as far as ESCO Scientific is concerned, we have another division, ESCO Medical, which is a separate entity now. And here we have the IVF incubators for ART, artificial reproductive techniques, and then ESCO Healthcare. So VaxiXL is the bioprocessing arm, and I'll be showing you some of the patented technology which falls under ESCO VaxiXL. And then we have ESCO Aster. So ESCO Aster is now a separate entity altogether, and we focus on offering vaccine. It's not only the product side or the equipment side. We also are in service. We are also a service provider as far as vaccine, cell and gene therapy development is concerned, or manufacturing services we provide to the bigger biopharma companies with our proprietary adherent tide motion platform. So that is one thing we will discuss more, a few slides on adherent tide motion platform. And then this technology, our patented technology, is supplemented by single-use suspension and fermentation system, and also downstream bioprocessing. So within the in vitro and in vivo diagnostic space, ESCO Aster, the right-hand side here, we just have a pointer here, a laser pointer, easier, yeah. So ESCO Aster on the right-hand side, you can see. So we also focus on in vitro and in vivo diagnostic space, where we develop monoclonal antibody antigens, single domain antibodies. And we also work with collaborators to provide turnkey solutions like mobile diagnostic labs, which during the COVID time, it was it's one of the hit product. It was one of the main products to supply. So ESCO Life Sciences Group, as you know, now we are a global company, and we are present in more than 40 locations in more than 21 countries all over the world. Distribution channel is much more than this now. So it's a little bit of old slide. So we are much more than 100 distribution channel globally, and we have our own subsidiaries, own companies in 24 offices. Manufacturing locations, main manufacturing locations still in USA, UK, Lithuania, Singapore, and Indonesia. So ESCO Astor, as I was saying, we are not only focused on the equipment side, but we are a service provider. We are a CDMO service provider. So in Singapore, we have our GMP facility, and head office is located in Singapore. So bioreactors. So bioreactors is a bioprocess tool. So we can define it as any manufactured device or a system that supports a biologically active environment. So you can see it's a vessel where we grow the cells, and it can be, or in other words, a bioreactor is a vessel in which the chemical process takes place, and which involves organisms or biochemically active substances, which can be derived from such organisms. So here what we are doing, we are growing the cells in vitro, and we are maintaining the parameters such as pH, dissolved oxygen, which is the aeration part, temperature, and also we monitor the culture medium, the nutrition of whether the cells are getting proper nutrition. So all these are monitored in a controlled fashion, and to grow the cells, we need a controlled environment in vitro, which mimics the human body. Right, so temperature has to be maintained. It has to be 37 degree, and all those, closer to the human body conditions. So we have broadly we have classified it. It's a classification by bioprocess scientists. It's like it falls into two categories: adherent cell bioreactor and suspension culture bioreactor. So we are very much focused on adherent cell culture bioreactors, which we have patent, and in the next slides, if you'll see that we have different types of bioreactors. So the broad classification was adherent and suspension. So today we will be discussing more on the adherent side. So there are many different types of bioreactors, as we can see in this slide, and including the stir tank, rocker, fixed bed, and of course ours, which is based on macrocarriers. So we have macrocarrier technology, which is on adherent cells. So if you can see the traditional multiple use bioreactors, they required a very high cleaning requirements after each cell culture run. After each cell culture, when they do the seeding, then they had to be cleaned. But the smaller glass bioreactors, which are glass systems, as you can see, the glass ones, so these can be sterilized in an autoclave. And the larger bioreactors, which were used, were mainly built up of stainless steel systems, and then they required additional plumbing in system for sterilization, such as cleaning in place and steam in place. So now the world is shifting, the whole bioprocess industry, whether it is upstream or downstream, we are shifting. There's a major shift from multiple use to single use bioreactors for obvious reasons. We will see the advantages. Single-use bioreactors were introduced into the biopharmaceutical market. It was somewhere around 25 years back, and they were made of single-use closed, gamma irradiated biocontainers. We also have our single-use bottles. We'll see in the next slide. So these were fitting on the reusable hardware. So hardware was static. It was multiple use. But then the actual cell culture, which was taking place, it was taking place inside the biocontainer. So biocontainers can be, it can be a bottle or a flask or anything similar in a cylindrical fashion. So it has to fit on a reusable hardware. So we also have a reusable hardware or a cradle on which the bottles sit. So single-use bioreactors these days are increasingly used for small scale, mid scale, and even large scale production, and they have many benefits. So we will talk about adherent cells here today, and you can see in the slides we have tea flask. This is the most commonly used in the labs, especially in the research and development. Then if the customer is scaling up, you have cell factories. If the scale up is more, for full production, even a lot of vaccine companies, even now, they're very much into roller bottles, which so our principle is a bit similar to roller bottles, and we are converting them one by one. Okay, so as far as application is concerned, you can see we have a lot of applications right from the vaccine productions to monoclonal antibodies, cell and gene therapy. So cellular therapy, as you know, it binds together. We have a cell processing isolator, and the customer is looking for a scale-up version from the flask, from the 2D system to a 3D system. We will understand what is a two-dimensional system of growing cells and what is a three-dimensional cell. So application-wise, cellular therapy is picking up very fast, and we have to understand, if we want to define what is a cellular therapy, basically it's a transplantation of the human cells to regenerate, replace, and repair the damaged cells or tissues, right? So basically, what we are doing in a cell therapy or cellular therapy, which is picking up very fast, stem cells, for example, every country have their now stem cell clinics coming up. So in those clinics, what is happening? They do transplantation of human cells. So these cells are basically to regenerate, replace, and repair the damaged cells. And at ESCO VaxiXL, we offer a tide motion platform that is very ideal in cultivating billions of anchorage-dependent cells. So adherent cells is also referred to as anchorage-dependent cells. For example, human mesenchymal stem cells. And depending on the type of vaccines, ESCO VaxiXL, we can produce very high titers. Like vaccines is one of the focus, as you know, now globally, now due to COVID-19. So we have a very good platform, scalable, linearly scalable platform for vaccine production, and as well as stem cells. Of course, gene therapy, and there are just a couple of companies involved in gene therapy globally, and Novartis is one of them. So yeah, so it takes a long time for getting the regulatory approvals. And then cellular agriculture, of course, it's a growing industry now, and we have certain success stories to share for cellular agriculture as well. So the heart of the system. So the heart of the system is BioNOC2. So just remember that BioNOC2 are the macro carriers, as I mentioned, that adherent cells. So as the name implies, as the term implies, adherent. So the cells need a substrate to grow. They need a base to grow. So in our case, the base or the substrate is a BioNOC. So BioNOC2 is a trademark from us. So it's a macro carrier. It's a macroporous carrier, and this supports the growth of anchorage-dependent cells or the adherent cells. So this can be for animal, mammalian, insect cells, and it can be for serum-containing or serum-free culture media. So basically, these BioNOC2s, they are made up of 100% pure polyester non-woven fabric, manufactured according to the CGMP guidelines. And they have very special geometric design. They have a very special geometric design, as you can see here in this slide. So the special geometric design helps and they have a special surface treatment also, these carriers. So this enhances the fluid mixing, and also it offers a protection from shear force, because low shear force is desirable in cell culture facilities where they do not want to, where the bioprocess scientists, they don't want to introduce shear, so that they get a better cell yield at the end of the day when during harvesting. So as far as the productivity is concerned, so if we are using a BioNOC2, so it is up to 5 to 10 to the power 8 cells. If we talk about the Vero cells, so we can do up to 5 times 10 to the power 8 cells, and the surface area is another thing to note here, that the growth area can be 2,400 cubic centimeter per gram of carriers. So in the later slides, we will see that the one bottle has 5 grams of carriers packed, 5.5 grams of carriers packed, and then 2,400 cubic centimeter per gram of BioNOC2, 2,400 cubic centimeter area we are giving per gram of BioNOC2. Then it's 100% animal component free, and the size is just 0.3 mm. 0.3 mm will be mentioned here. Yeah, it's just 0.3 mm thickness. So the thickness is just 0.3 mm. This is for easy exposure to culture medium, which aids in the oxygen transfer. So the BioNOC2 is a non-pyrogenic and not toxic, and it's biocompatible. So we also do special surface treatment before the customer uses BioNOC 2. So this is to enhance the biocompatibility of the BioNOC 2 carriers, and these are compressible for very high cell density, and the hydrophilicity is a term very commonly used in bioprocess materials. So hydrophilicity is like the water retention or the water attachment to the BioNOC 2 is it's very stable. It's for 4 years, and these BioNOC 2 carriers are sterilizable by autoclave. That's one of the questions very commonly asked by customers. So these are autoclavable. These can be gamma irradiated and ethylene oxide sterilization, and it offers a very low lint, which is very convenient for when compared to the other macro carriers on which the cells grows. Our BioNOC 2 has a very low lint, and it's convenient for adherent cell growth, whether it's for protein or viral protein production. Yeah, so this was a very major slide. As you can see, now the next questions from the customers will be, okay, fine, BioNOC 2 is there, you have a good macro carrier, then which cell lines can be cultured? So basically all major adherent cell lines, whether it's Vero, mesenchymal stem cells, insect cells, SF9, all this can be cultured. These are also on our website, so you can feel free, or if you are not sure, just send us a message or an email. So these are the cell lines which can be grown using our tide motion principle. So I have a short, I have a clip here. So
got no sound.
we have our own ESCO channel on YouTube. So if you just type ESCO on YouTube, you will get our channel. We have more than 50 videos there, product videos as well as demonstration videos. So feel free to watch that. So tide motion principle. Now coming to the core tide motion principle. So the term tide motion, what does it imply? So the term tide motion is derived from the cyclic high rise and the low rise bodies of water on Earth. So in the bioreactor term, what happens that if you can see this bottle, again, I'll take the laser pointer. So if you see these bottles are all single use, and we have 4 placed on the benchtop bioreactor. So if you can see in the bottom portion here, we have the red colors. These are the culture media. And so now the question is, where are the carriers? So on the middle portion of this bottle, these are the vented bottles. You have the vents on top. So in the middle portion, the carriers are nicely packed in the middle portion, the white color here. And what happens in this whole principle that we subject these carriers, the cells are growing on these carriers. So we subject these carriers alternatively to aeration. So once the bellows, we call it bellows. So you have 5 bellows here, 100 ml each. So that is 500 ml of culture media. The red color is the culture media here. So once the bellows are pressed, and the pressing of bellows is through pumps located on the cradle. In the next picture, you'll see that we have a stainless steel cradle which has pumps. So the pumps will press this bellows upwards, and the culture media will climb up in the bottle, will rise up in the bottle. And this will completely submerge the carriers here where the cells are growing. So the cells get food, that is nutrition, at one point of time. And once these bellows are released back to its original position, then you have vents on top. We have a filter and vents on top. So the air will gush into, will enter into the bottle through these vents, and then the cells will receive aeration or oxygen. So it's a very simple process. We are subjecting the cells one time alternatively to nutrition, that is their food, through the culture media, the red color ones. We have 500 ml of culture media here, and then the cells are getting oxygen. So we are giving them oxygen as well as food. Two important things for them to grow. So that is the main principle. So in our bioreactor, there's a very gentle upward and downward motion. As you can see, we don't have any stirrer mechanism or anything to introduce stress in the bottles. So this is very gentle upward and downward motion of the culture media, the red color culture media, and basically for adherent cells, and the nutrition exchange happens. So this submerging and emerging principle allows the cultured cells to produce quality products that are always of very high yield. So we can get a very high yield because the cells are not traumatized by stirring mechanism or any other friction or something. So we have a very gentle movement of culture media up and down, and that's how the cells grow, and they are of very high yield. And if you notice here, there's no oxygen limitation. So dual oxygenation action, because once it has practically no oxygen limitation, and regardless of bioreactor scale. If we are taking a small scale to pilot scale or to bigger scale, in the next few slides you will see. So we have no oxygen limitation, and there is no bubble formation, as you can see, because there is no bubble formation because the whole media climbs up and completely submerges the carriers. So there's no bubble formation. So tide motion bioreactors, because in the larger scale from 2 liter onwards, we use separate vessels for culture, and even the stirring is done separately. So this isolates the cells being cultured from any form of bubble or foaming. Okay. Then this slide is depicting our linear scalability. So we start at a very small scale. As you can see, we have the cell cradle here. So cell cradle is the cradle, and then we have the 4 bottles on top, which are disposable. And even we have a smaller than that for proof of concept. If the customer says, okay, fine, we want to try on a very small scale in our lab to determine if the cells are attaching and growing properly, then they can use a Cell-X rocker here, which mimics the tide motion principle. So it's the smallest version, basically for proof of concept. Then moving on, we have the cell cradle and with a shaker, and then we have cell cradle X, which is automated unit, and then from 2 liter onwards we have tide cells. So we have a very linear scalability shown in this diagram, and we can go up to 5,000 liters in packed bed. So from a very small amount, 500 milliliters to 5,000 liters, we can go on the same technology. And this is desirable by all the bioprocess scientists and researchers. So the ability to scale out or scale up is one of the key advantage of tide motion bioreactors. And as you can see, we conform to CGMP and GMP standards, ISO 13485. And we have several configurations, whether the customer is opting for single use, autoclavable, or with SIP capability, we have the solution. So tide motion bioreactors, as you can see, we have the lab scale, pilot scale, and full production. So the shortest bioprocess scale development time. We offer the shortest bioprocess scale development time, and there are certain parameters developed in the academic or industrial R&D, which we can easily translate into clinical trials and commercial productions, as the parameters are identical, because nobody wants to use a technology in the lab scale and then pilot he has to use another technology, because they have to go through regulatory agencies, and it's very time consuming and expensive, right? So these are the different scales. So we start from lab scale, pilot scale, and we can go up to full production. So Cell-X rocker is the smallest device or equipment we have for cell seeding. So here we can do up to 20,000 to 60,000 cells per carrier to show as a proof of concept. So this is our device. So basically it's just a simple 2D rocker, but we have customized it. We have customized it for setting up the top holding time and the bottom holding time. So it's something like this, so in simpler. So you have T-flask here. So in this T-flask, you can see the white color ones are the Bionoc 2 carriers on which the cells grows. So once we are providing, here is aeration and here is nutrition. Nutrition meaning The cells are completely submerged, the carriers are completely submerged in the culture media. The red color is the culture media, and once the flask is tilted this way, then it gets the culture media is here and it gets oxygen. So same thing, we are subjecting the carriers alternatively to nutrition and aeration. Then cell cradle. Cell cradle is our blockbuster product. We have several references globally. So we have two models here. One is the batch system, which if you are aware of CO2 incubator, it needs a CO2 incubator. So it fits very nicely into 170 liter CO2 incubator. And then we have the recirculation system, which is also known as perfusion system. So perfusion system, you will need 240 liters of CO2 incubator. So that's how we club together our CO2 incubator and the small benchtop bioreactor. So all these bottles are disposable. They are single-use bottles, and they offer very high cell density. And these are single-use, cost-effective. We will see a comparison between 2D system and the present system in the market. So this is one small clip to play for cell cradle. Okay, so what are the different components in cell cradle perfusion system? So we have the cradle here. Cradle is your stainless steel, which is a fixed component here. This is the fixed component. Then you have the 4 bottles on top, and then you have the feeder bottles and cell feeder pumps for feeding the cells. So we have 4 pumps over here, and these are the bottles. These are the consumable bottles, and these tubings are also consumable. So the bottle and the principle, the same thing. We are subjecting the cells or the carriers alternatively to aeration and nutrition. You can see we have the cell seeding. So we have 2 components here. One is the cell seeding, where you grow the cells and then you harvest the cells. So both these videos are present on the YouTube. For details, you can just log in and see our cell seeding and harvesting videos. So we have a controller which sits outside the CO2 incubator, so it has magnetic legs, so it sits outside the CO2 incubator. Then you have the cell feeder pump here, so you have 4 peristaltic pump here for cell feeding. We have an automatic cell feeder now, and this is designed to simplify the process, simplify the operation, which was a bit manual on earlier on, but now we have a generation 2 cell feeder pump, which is automatic. And cell cradle, the whole system is integrated. It can fit inside a CO2 incubator, as I mentioned. So where the pH and the temperatures level is controlled. So and the sampling can be done when the scientists want to see that, okay, we have to do sampling to see if the cells, for example, stem cells are growing or not. So they do some sampling. So we have a sampling port where they can use a forceps and they take out the carriers from different areas of the bottle, and then they place it in a petri dish and check the cell growth. So this is the mesenchymal stem cell growth after 7 days. So as observed under a microscope, this is how the stem cells will look like. So the cell cradle is a very simple device. It has just 4 parameters to check. So you can see this inside this cradle, you have the pump. So the pump presses the bellows, and the liquid, the culture media will go up and submerge. These are the pink color ones are the the carriers. They were submerged under culture media. That's why the color had changed. And then you have the vented. These are the vented caps. So these bottles are vented, and you have a filter on top. Right. So only 4 parameters have to be controlled through this controller, which is outside the CO2 incubator. Bottom holding time. So this is the time required for amount of, this is for the aeration part. Then you have the top holding time, which is the amount of time for nutrition. Then you also control the speed. So basically what you are doing, you are controlling the holding time of this culture media, and also the speed at which they move up or down. So one bottle. So now coming to the thing that how we compare to other technologies in the market, which is mainly 2D. So our bottle, one bottle is equivalent to 10 spinner flask or 100 petri dish or 20 roller bottles. So this is a very powerful slide to show to the customers. It's also on our website. So you can imagine that the amount of culture media needed for our bottles or our technology is far less than the competition. And the customers are always looking for a return on investment or the capital expenditure or the operating expenses. So when we show them that, okay, look, if you are using one bottle, these others are also disposable method or single use. If you are using one of our bottle, you are saving a lot in your culture media, and also the time and the manpower requirements. So we have also launched a shell checker for harvesting. Once the cells are seeded, then it will do the automatic harvesting. So as you can see, in tight motion bioreactors, the CellX rocker is the smallest. It's just used for beta testing macrocarriers in the T flask on a very small scale here. And this it's basically a 2D rocker. So this provides very gentle side-to-side rocking motion for mixing in culture flasks. It can be tubes or even dishes. And the rocker is designed to conveniently fit inside most of the CO2 incubators and refrigerators. So therefore, the assay can be placed in a controlled environment. So CellX rocker is the smallest one used for proof of concept. Then moving on to the cell cradle. So cell cradle is a disposable bioreactor. We call it single-use bioreactor. So where the cradle is fixed, that is permanent fixed, but the bottles are disposable. And it's capable of very high density cell culture for protein expression, virus, or monoclonal antibody production. And so it's designed mainly on the concept of bellow-induced flow of media. As you can see, we have 5 bellows there with 100 ml of each, 100 ml media. So it provides a very low shear, high amount of aeration, that is oxygen for the cells, and foam-free culture environment. There's no bubbles. There's no bubbles and foams. In some of the bioreactor technologies, still they are not able to solve that because it's a limitation of the technology itself. Then Cell Cradle X, the third one, the third stage here. So Cell Cradle X is a single-use, closed automated bioreactor. This is also based on very much on the Tide Motion platform, and it's manufactured in an ISO 13485 factory, and it complies with CGMP requirements, which is also a compliance to 21 CFR Part 11. So this is a very we can say it's a semi-auto, and it also comes with a semi-automated harvesting system. Then from 2 liter onwards, we have Tide XL. So Tide XL bioreactor is the extended large-scale system. It's large-scale system. You see, we are linearly scalable. So this diagram or this picture I have to show and also to the customer so that they are rest assured that they have confidence in the technology, that we can use this from a research and development stage, then slowly scale up. So Tide XL 2 liter onwards is the extended large scale system for the of the lab scale cell cradle system. So cell cradle is here, and then you see now we have 2 liters here. So in this system, what happens that we have a matrix vessel where we pack all the BioNOC2 carriers, and that also function as an artificial lung with a very huge surface area for it provides a very huge surface area for nutrition and aeration. So we have discussed about Cell Cradle X now. Cell Cradle we have seen. Now coming to the Cell Cradle X. So Cell Cradle X, as you can see, is a very integrated system, standalone system, single-use bioreactor. Here also you can see the bottles are the same, and of course for the same applications. So it has also single-use sensors, whether it's a temperature or pH, and it also comes with a glucose and biomass sensors, which are optional. So let's see a Okay, so the next is the Tide XL. So Tide XL is used for producing stem cells and viruses in manufacturing scale. So Tide XL from 2 liter onwards, we already have customers in commercial production who are producing stem cells and viruses. So the expansion process for both, if we talk about stem cells, because I'll be speaking mainly on some key segments and applications. Of course, we have more than 10 applications as of now, but the main ones still are stem cells and vaccine production and virus. So the expansion process for both the stem cells and virus from the 2D systems, which is available in the market, is troublesome, because these 2D systems are enough for research and development scale, but when and it caters only to a very small number of patients. If they are using Flask or even cell factories from some companies, so it can only cater to a very small number of patients. But as these applications evolve and they need billions and trillions of cells, so they also need bigger systems. So they need bigger systems to achieve high amount of virus and cells, and that is how we come into the picture. And of course, with the adherent cells, when they want to culture adherent cells. So our ESCO VaxiXL Tide cell, Tide Motion bioreactor, is designed to produce very high viral titers through our own proprietary 100% media exchange, and we can do 90% more than 90% stem cell harvest. We have our data, and it's also, we have some white papers on our website for your perusal. You can also share them with the customers. So our Tide Motion principle is also very flexible. It offers cells with 3-dimensional in vivo environment. It has very low shear stress and high productivity. So for example, if we are using a 100-liter packed weight volume, so we can simply replace 100,000 pieces of Flask. So this again saves labor, time, and space requirement for the customers. So Tide XL is all these units which we are seeing from 2 liter onwards. They are all available in different configurations, and both single use or multiple use. Automated controls equipped with the pH, dissolved oxygen sensors, and sampling ports for cell monitoring and observation. So from 2 liters onwards, it's completely 100% monitoring. So very less human intervention is there. So this is how the Tide XL looks like. So the same bottle is now into 2 parts here, as I mentioned. So we have the matrix vessel and the mixing vessel here. So mixing vessel is outside, and the cell growth takes place inside this incubation system. So the smaller systems we have to put inside a CO2 incubator. So the bigger unit from 2 liter onwards, there is no need for a CO2 incubator. We have the incubation system here integrated, built in. So we have a built-in incubation system. We have a Siemens HMI system here, and of course complying to all the international standards. We have the pump. So the cell culture still takes place inside. The vessels are all configurable. We have a table to show which capacity we can. So these are the components. So the TCISCS, this is the complete incubation system, as you can see, and the matrix vessel is inside with a BioNoc 2 carrier. So the cells are growing inside the incubation system here. We have a mixer outside with a heating jacket with all the sensors here. We have the pH, DO, temperature sensors here, Siemens HMI, and as you can see that this mixer or the mixing vessel, this controls the temperature of the culture medium, which is very important. So this is the incubation system, same. I have a video to show you. Yeah. So moving on to the matrix vessel options, what we have. So 2 liter onwards, as I mentioned, we have multiple use, single use, 10 liters, 20 liters, 100 liters. So far in commercial production, we have supplied up to 100 liters. So as you can see, the matrix volume, depending on what kind of like 1 to 2 liters for multiple use, single use 2 to 4 liters. So these are available in glass as well as polypropylene PP, which is single use. So yeah, then coming to the now we have seeded. So we have grown the cells. How about the harvesting system? So we are one of the companies in the world in bioprocess having a completely 100% automated harvesting system, cell harvesting system. So the whole harvesting system runs on a setup from washing to cell harvesting. We can do in a closed automated manner. And this our harvesting system is very much suitable for cell harvesting from Tide Excel 2 liter matrix vessels to 20 liters, 100 liter vessels. And we support this particular system supports the cell seeding source for Tide Excel up to 100 Tide Excel 100 system, which is 100 liters. And the harvesting viability of animal cells, which we can do up to 1 to 3 hours. And the main thing to note for our harvesting system, it works on 2 principles. We have a mechanical agitation. So we take the vessel, matrix vessel here on a horizontal fashion. We place it inside this is in equipment, and then the system will give it a horizontal. It will give a horizontal mechanical agitation. So that's one. And then the second part is dissociation enzyme. So it works in 2 steps, and up to cell harvesting efficiency also depends on the cell line used. So we have some data, and we have videos on our website and also on the YouTube. Feel free to look at it. And then if you have any questions, so please ask on our harvesting system. So just to let you know that we are a pioneer in this segment, and then we have a completely 100% closed automated harvesting system. So as I was mentioning that matrix vessel has to be placed here in a horizontal fashion. Normally for dissociation enzyme, we use trypsin here. So you have these harvest pumps, you have the culture media pumps. So this is how there are 2 components. One, you have the installation of the matrix vessel with the mixing bag is placed here, and then you have to rinse the matrix with PBS. So these are the 5 steps, and your harvesting is done. Just, it's fully automated. No human intervention is involved. Yeah. So now comparing T25 flask to our 100 liter matrix vessel here. So you can just see that 800,000 of T25 is equivalent to 100 liter matrix vessel of Tide Excel. So this is a very powerful slide to show to the customers that you see the amount of culture media, time, and manpower needed. And then with just 100 liter of matrix vessel, it's just equivalent to 800,000 of T25. So there's a huge saving for the customers. So we have seen three categories of products for bioreactors based on tide motion principle: cell cradle X, and tide XL. So this table summarizes the different specifications and whether and also on the regulatory compliance. So cell cradle, as I mentioned, it's for the research and development and on a smaller scale. But 2 liter onwards, or even cell cradle X, it complies to 13485 ISO standards and also United States Pharmacopoeia standards 1043/665. Yeah, so our solution is scalable from seed preparation to the transfer. It's all done in entirely closed system. And if the customer is interested, we just combine it with our cell processing isolator, and then they get the complete solution. Some of our customers are already using our bioreactors and also the isolator system. So when compared to the 2D and 2-dimensional and 3D cultures of mesenchymal stem cells, as you can see, so if we use 3D, our percentage of stemness is 98%, which is quite high. And we also can get a very large number of cells depending on 1 ml of media. So you can see cell number and media usage. So if you use a 2D, if the customer is using 2D, they'll spend they can get 166K cells for every 1 ml of media, and we can do 400K cells for 1 ml. So there's an incremental cell number growth and using the same amount of culture media. Then comparing to the other technologies in the market, whether it's a stirred tank, static or rock method or hollow fiber method, ours is emerging and submerging. So all these blue colors are the advantages what we have over competition. So we have a very small footprint, as you can see. People have to use hundreds and thousands of flasks, and then we want to replace them, and we are replacing them fast globally. And the rest of the method, as you can see, if the customer is using a cell factory, it's very manpower intensive and laborious. And in certain conditions, like if the customer is using a stirred tank bioreactors, those show the shear stress is very high. Foaming and bubble formation is there. And then overall, they have oxygen limitation, and linear scalability is a big issue if a stirred tank is used. But in our case, all these blue colors, so we have several advantages over most of our competition and other technologies available in the market right now. Then moving on, this is one parameter which is checked in the lab on a daily basis several times. So this is glucose concentration. So we have a glucose meter. So this glucose meter is similar to what you have for blood glucose measuring device or a meter. So our glucose is like a complete package. It's a portable meter with disposable strips. As you can see, we have the disposable strips over here, and it's suitable for glucose measurement for both serum as well as serum-free culture media, and it can be used for both mammalian as well as insect cell culture. It's very small, just 70 grams, and fits perfectly in your pocket. And the compact size allows measurement to be done right next to the culturing device. The customers can place just beside their bioreactor and conduct a lot of tests. So it's a 25 strips per bottle. So 2 bottles are provided in the package. So you have the glucose meter plus the 25 each. So it's 50 strips in the package. So we have a short clip. I know that the sound is a problem, but it's okay. At least you get an idea that how the product looks like, and you know. Yeah, so the Glucell meter is like, yeah. So the Glucell meter it has a very portable design, and the interface of the Glucell meter is very intuitive, and it's very easy to use. So there is no learning curve, very steep learning curve. All this is not required, and the meter can store up to 180 test results with corresponding time and date. That's very important. And then recording of important data in the lab is very convenient, and it comes with all these expiry dates and everything is mentioned on the strips. So it's super easy to use. So we recommend all the labs, and it's and also our partners, if you can think of, we can work out a package for you. It's very easy to sell, low cost device. And of course you have the bioanalyzers in the market which are like few $100,000. So they do a lot of other parameters, but the most important one is glucose concentration for the culture media, so that they have to check, the scientists have to check on a very regular fashion whether it's still the nutrition is intact or not. Okay, so now coming to the segments where we have to look for a customers for bioprocess. So it can be academic research institutions, CMOs, CROs, contract research organizations, clinical industry, life sciences, and of course bioreactors falls into two categories. So we are very much into adherent cell culture, and then these are the applications: vaccine development, cell therapy, cell and gene therapy, bone regeneration. Of course, in Europe we have some good traction in bone regeneration. Artificial meat, yes. In Netherlands and other countries in Europe, you'll find there's a very big potential going on, and a good amount of funding also. VC funding in artificial meat, and then of course in proteins and others. So the basic question, the first question to ask the customers or the client is, what is the cell line they are using and what is the end product? So depending on, because as you know, we are very much focused in adherent cell culture, but of course we have hybrid models which can do both. But that's not a very focus area for us. A tide motion principle is the focus area, and for that we are aiming at adherent cell culture. Then we have to understand what's their current system, whether they are using 2D methods or they are already using a 3D, whether it's a hollow fiber, etc. Then we have to ask them the third question, that how many current systems they have and what's the target scale, and the timeline is very important because most of these projects are funded externally and they have budget and timelines and to achieve. So they will mention, most of the time they will also mention on the website that look, we are into the research phase, and we will after 2 years we will scale up or looking to scale up because they have been given timelines by their investors. Most of the companies, whether it's a vaccine company or cell and gene therapy, these are not self-funded, so they have a timeline to meet. So the customer engagement model is very simple. We have questionnaires on our website, and upstream questionnaire is quite general, so that you can provide to a team, and we will be able to find a model for you, and then we interact with the customer. So most of the time it's like the pre-sales part is quite simple, and and the rest of the customer interaction and scientist to scientist interaction will be taken care by ESCO Aster team based in Singapore head office. And this is some of the references we have globally. So we have now ESCO Aster VaxiXL map over here. So we have quite a number of units already. So with this, I conclude my presentation, and I hope you got an idea of which technology we are talking about. Cell processing isolator is very clear from Alvin what we have. And then for bioprocess tools, we have bioreactors, and it's a patented bioreactor where if you are bidding in a government project, you are going to win 100% time because it's a proprietary item, it's a patented item. So there will not be another supplier based on the same technology. So tide motion, 100% you will win if you are going to a research center or a government institute. And the private companies, why they will be attracted to us? Because they will be doing a very significant saving on their OPEX and CAPEX and OPEX financial ratios. So with that, I end my presentation. If you have any question, please send them on chat or send an email. Our email is mentioned here. So you can visit our website. ESCOaster.com is our main website. And if you want to know more details on VaxiXL products, then we have ESCOVaxiXL.com. That's more on the bioprocess tools. Yeah. So with that, I end my presentation, and I thank everyone for your time today. Yeah. Yeah. Thank you. Dimitri. Yeah.
Thank you, Ahmed, for your presentation. Once again, dear partners, thanks a lot for participation on this event. I hope that you will have some questions after you digest a little bit the information you get today. And we appreciate if you share the feedback via chat or via form, which we will try to submit to you as a follow-up for this training. Okay, ladies and gentlemen, thank you once again, and we'll keep in touch. Stay healthy, and hope to get some feedback from you on this topic. Thank you, and bye.
Thank you very much.